120 employees dedicated to building and supporting our product, half of which who support customers directly or build the product itself. So for those of you who don't know, benchling has a suite of biologically intelligent applications. We have a molecular biology tool, which we're going to dig into in great detail today, that allows you to do DNA and protein design, as well as a notebook that allows you to capture protocols and record data and results for your experiments. We have a registry system that allows you to not only store your DNA and proteins, but other pieces of data that you would model, such as cell lines, viruses, containers, refrigerators, freezers. We have an inventory tracking system that will track the physical manifestation of all of these samples in your lab.
Okay, thank you very much, Ashoka. So I'm now just going to jump into the benchling platform to begin our demonstration. So before we kind of get into the agenda for the demonstration, just a couple of things that I'd like to address. So the first is that Benchling's a cloud platform for tracking all of the biologics that you work with at the bench. When you're using any of the 6 Benchling applications, you'll notice that I'm actually just in the browser in Google Chrome, for example. So all of the work that you'll be doing is just simply through your browser. You might also notice that I have opened up a Benchling Lab notebook. So the vast majority of today's demonstration will kind of be coordinated within this ELN entry that I've created. Awesome. So let's just briefly take a look at our agenda. So we're going to spend a little bit of time showing you how you can bring in your vector NTI archive files into Benchling. And then we'll just transition to the bulk of the time, which will be spent on this lentiviral plasmid production demonstration that Ashoka just detailed. So let's begin with this first part, bulk import of vector NTI files into Benchling. So again, today we're focusing on maybe a vector NTI archive file such as the MA4 files, but I just want to iterate that you can actually bring in a variety of different file types that are supported in Benchling. And when we mean that they're supported in Benchling, what we're saying is that not only the sequence, but the annotations and tags associated with those sequences will carry over into Benchling. And so it's included but not limited to the file formats that you see here. For today's demonstration, however, I will be bringing in an MA4 archive file from vector NTI. So you could imagine the typical workflow for this would be, you download or export your archive file from vector NTI. You then navigate back into your Benchling browser. You find a particular project where you'd like to store your files. You can see that I have a couple of subfolders nested here, including one for today's demonstration. Now, for me to bring these vector NTI sequence files into Benchling, I can simply click on the Create button and choose Import DNA Sequence. With one simple click and drag motion, I'm able to bring in all 11 of the sequences contained within my archive file. Taking a step back, I am a scientist that's working with the vector design group or vector design team in my lab or institution. And a scientist is going to submit a request for me to design and then synthesize their lentiviral construct at the bench. So to begin with requesting the design and synthesis of a lentiviral construct, you could imagine that the scientist first designs some sort of shRNA oligo. They follow the criteria they need to design these oligos. These oligos will ultimately be used to knock out some target gene in a cell line. You could imagine that the scientist could actually come into Benchling and utilize something like Benchling's registration tables, which is an extension of our registry application, to register the shRNA oligos that they've designed. So with a couple easy clicks, they're able to register these shRNA oligos that they've designed so that they can, reference them later down the line and prevent any redundancy from people recreating those fragments. Using Benchling's requests systems, the scientists can actually initiate a request for me to design their lentiviral plasmids directly from the registration table. So for example, they can create a request for a lentiviral plasmid synthesis. They can fill out important metadata like the day they're requesting for this to be designed, the assembly method that they'd like me to use, for example, Gibson assembly. They can even specify things like a vector type that they'd like me to use, for example, the PLK01 backbone. When they create this request, me, the vector design scientist, will receive a notification that this has been assigned to me. I can go and find the proper metadata that I need to do my assembly. And what this does is essentially prevents this kind of disparate system where a scientist has to either email or message or go find another scientist in the lab, ask them to fulfill this request to design this plasmid, hand over the notes that they have, and centralizes it in one place here. Okay, wonderful. So you can imagine the scientist then has registered their shRNA oligos. They've submitted a request using Benchling's request system for me, the designer, to go and build these plasmids. So before I can go to the bench and actually synthesize the vectors, I need to use Benchling to build my vectors. So in today's demonstration, we'll be using the bulk assembly tool to insert one common promoter in combination with 10 unique shRNA oligos to generate 10 output lentiviral plasmids. And again, we're going to do so using Benchling's bulk assembly tool. So I can first start by navigating to the blue navigational toolbar on the left-hand side to this Create button. From there, I can hover over DNA sequence, and I can actually just choose bulk assemble DNA. This then navigates you to Benchling's bulk assembly tool. The way that the bulk assembly tool works is it's kind of this, like I mentioned, a combinatorial plasmid generator, which allows you to create really complex molecules in bulk with just a couple of simple clicks that are supported by display features and things like that. So I can first start by selecting the fragments that I'd like to assemble. For example, in this case, you might remember that the requesting scientist asked that I use the PLK01 backbone. And then the next step is for me to select a fragment end. Another way that you can think about this is selecting your assembly method. So currently in Benchling, we offer a variety of bulk assembly or assembly methods. Enzyme-based would be, of course, if your fragment was flanked by restriction enzyme cut sites that will produce some sort of overhang. We have homology for quite literally Gibson assemblies, for example, where your fragment has some homology region that's conserved between it and the backbone. And then finally, we have concatenation. So concatenation is an assembly method that allows you to piece together your fragments into a plasmid without it representing any specific technique or experimental technique that you're using at the bench. In this case, the requesting scientist asked that I do a Gibson assembly to design their plasmids. And so I can pick homology as my assembly method. I can then type in my homology regions, for example, TG, 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 GC, 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 GC. You'll see that the last two steps, selecting fragment size and direction, are now supported by display features that appear on the right-hand side of the workspace directly underneath Backbone Summary. I can choose to keep either the larger or the smaller fragment. I can change the orientation of my fragment if I so choose. And again, these last two steps are supported by these display features that we see on the right-hand side. The next step is for me to add my promoter. To do so, I'll click into the Insert One box and follow similar steps to what I just did, navigating through Benchling's tree structure, locating my promoter, entering in my homology regions. And what's fantastic about Benchling's bulk assembly tool is that it automatically remembers the homology regions that we had selected in the previous window. So I can simply click on my previous homology regions I can add in new homology regions if need be. And again, you can see that this last two steps, fragment size and direction, are supported by these display features on the right-hand side. The last step is for me to select information against the promoter itself. So now that I've designed my vectors, my theoretical vectors, you could say, I've annotated them or tagged them so that I can reference the pieces at a later point. The next step is for me to actually go to the bench and synthesize these vectors, right? So what I have displayed here for you is an assembly protocol, right? In this case, as the vector design scientist, I might have already created a PCR fragment or a fragment that contains the homology regions that I used in my Gibson assembly. I can actually mention those fragments here to create links to them so that I can reference them later down the line. You'll see that when I click into this, I can see any relevant information about the primer that I designed in Benchling's primer tool, as well as all of the particular fragments or the particular vectors that this has been attached to in the past. So quickly referencing the primer and its associated plasmids is as easy as clicking into the @mentioned primer. I then might do something like run a 2% agarose gel and gel extract my DNA fragments. If I've taken an image of my gel, I can attach that into Benchling's notebook here. I can go back to the bench and combine my fragments, following some sort of standard Gibson protocol, for example, combining my fragments in equimolar concentrations. Take the next steps to do a classic transformation of my plasmid DNA in E. coli, such as DH5 alpha. I can plate that on an agar plate, pick my colonies. I can amplify them in something like LB broth, and then ultimately use some sort of kit, for example, to obtain the plasmid DNA associated with the lentiviral construct that I designed. In similar fashion, you could imagine as me being a plasmid vector design scientist, I might want to register these plasmids now that I've gone to the bench. I've actually created them. I've captured specific data against them. I might want to, again, leverage Benchling's registration system to capture and monitor these vectors down the line. Actually, Benchling's inventory application integrates with the molecular biology suite and the registry as well. So I can use something such as a transfer table, which is an extension of our inventory application, to actually directly add the samples that I've recently created. For example, the plasmid entities that I've understand which sequence that you've made as your template, will create the alignment for that particular template and save that alignment to the template sequence. So you'll see here that we've now created one of our alignments, and you might notice that there's a lot going on here. And this is because our alignment tool offers an abundance of very, very interesting, unique display features, all of which can be toggled via display icon, similar to when you're looking or viewing a sequence map itself. I can look at specific reading frames. For example, I only want to look at one reading frame. I can drag across this mini map here to find any of the mismatches that appear in red or any ambiguous bases that appear in yellow. I can even show a compact view if I'm working with a large number of sequences and I kind of want to just see the mutations, for example, or the differences between my AB1 files and my template alignment. And so you can imagine doing something for a much larger alignment, something that might look like this, where you're aligning 30, 40, 50, 60 sequences in one alignment. And this allows you a more global view of the samples that you prepped on the bench compared to the ones that you've designed in Benchling. So you can see now in a matter of 28 minutes, I was able to design a number of plasmids based off of requests that a scientist has made to me. I was able to annotate or tag my sequences with the components that are, within my particular plasmid. I was able to promote those fragments to the registry where I could capture critical metadata against it. seamlessly attach sequencing primers that I had designed in the past to the vector that I've created, design new primers in bulk against a particular sequence, bring my AB1 files in and create an alignment without ever having to leave the